Thank you very much for the invitation and for the organizers. I will be talking about cellular crosstalks as a potential tool or product for treating cancer. As you know, the smallest unit of life is the cell. The cells, they are either animal cells or plant cells or bacteria, and these cells, in order to work or to function or to survive and proliferate, they have to know first of all the environment where they live, and also they have to communicate. This is very important for their homeostasis. This is important to maintain the stability and continuously adjust to the surrounding conditions necessary for their growth. And this is what life means. Life means to grow, to reproduce, and to function. So I actually said communication. Communication between cells is so important for their survival and for their growth and reproduction. The communication between cells occurs between plant cells, animal cells, and bacteria. And we can see that if the cells, they are in close connectivity, they actually like they secrete some bioactive molecules, and this is for the local communication. There is some other long distance communication between cells, like secreting hormones and affecting other cells. For example, cells in the pancreas secreting some hormones that they are affecting liver cells and adipose tissue cells and also muscle cells in order to do uptake, for example, for the insulin uptake for the glucose. But the most important communication between cells is through vesicles, extracellular vesicles. These extracellular vesicles, they are secreted from cells and they go to the recipient cells. They have a cargo inside. There's lots of material, lipid, proteins, messenger RNA, and they have their effect on the recipient cells. These are in my talk, I will be talking about extracellular vesicles, or what's called in the literature exosomes. Exosomes, they are mediate the exchange of information between cells. They can trigger or stimulate some chemical or biochemical pathways, and also they can supply some material to the cells. For example, in here we can see that there is a direct stimulation, like providing some material, some information, or triggering some stimulation. Here we can see that the cells, they can provide, for example, receptors. If the cell needs some receptors, if the cell needs some proteins, they can provide it with some proteins. Also, they can provide with genetic material, microRNA, RNA, and also double-stranded RNA. This communication occurs in health and in disease. So cells, they can, as I said, they communicate between them. If they are healthy, they would like to communicate and function and like do the homeostasis between cells. However, if there is a cancer cell, which is a disease cell, they also would like to transfer their disease to the other cells. they can communicate through these exosomes. If you write an exosome in the PubMed, you can see that there is lots of literature. The active, this is a very active research field. Like in 2013, there has been a major increase in the literature, and also in the 2022, there has been lots of publications, about more than 5,000, and this is indicate that there is lots of activity in this field, which is means exosomes. Actually, at 2013, there has been a Nobel Prize for two distinguished scientists from California that have discovered the machinery regulating physical traffic inside our cells and also inside other cells like plant and bacteria. And from that time, there has been lots of work on these exosomes. However, there has been lots of improvement in the how the biogenesis of exosomes. As we can see here, cells. This is a cell. We can see here that this is the membrane of the cells. First of all, some invagination occur in the cell and leading to the production of early endosome. Early endosome try to exchange some material between the endoplasmic reticulum and the early endosome, and then they develop to late endosome, which start also exchanging some material with Golgi, and then some production of multi-visible body has been produced here, and they can move through the cytoskeleton of that cell intracellularly and is secreted outside. If it's not secreted outside, it can go to degradation and recycling again. These exosomes, which are, as I said in the beginning, they are bilayer of membrane. They have the membrane of the cell, but they don't have any organelles inside. But they do have lots of material or molecules. They have proteins, they have enzymes, they have RNA. About more than 400 RNAs. They have DNA double-stranded and single-stranded, and also they have some proteins on their membrane. This is important for the adhesion of the cells and releasing their cargo inside the cell. Something also very important with these organelles that they are new. They have lots of heterogeneity from their size, from their content, cargo, and also from their functionality and their source. Like these exosomes, they are secreted by all cells in our body. We can find them in the plasma, we can find them in the saliva, in the urine, and we can find them in all our body, and we can even isolate them. And then 5 minutes, okay. So this is the exosome. Exosome is, as I said, have integrins for binding with the cells and releasing their inside cargo, and have tetraspanine. These are just specifying the cells and binding to them. Exosome means just like an email. Cells send exosome to other things and try to affect them. There are some characteristics of these exosomes that I will not go through all of them, but they are very small, about 100 nanometer in diameter, and they have removing and transmitting some material to other cells for the homeostasis. And we have said, this is very important thing that exosome can promote or restrain some disease. And this is from my research has been started from there. Like we have chosen some tissues from our body that we know that they don't get any cancer, or the cancer there is very rare. So we can take these tissues and isolate their exosome from there and test their anti-cancer activity. And also this is, I will go through it. This functionality is very well known. It's important for the proliferation, for development of cells, waste removal, but this is important for the growth and the homeostasis of the cells. So what was our strategy in our research was to locate and isolate anti-cancer extracellular cells from some tissues, and we tested them in vitro and in vivo on cancer cell lines. And also we could use them, or we are planning to use them, these exosomes, as a delivery for some materials. So this strategy is not gene therapy, and it's not cell therapy. It's using exosomes that they are secreted by our cells. So isolation is just by ultracentrifugation, and we can characterize them by nanosite and electron microscopy. And we have recently isolated them from two tissues, from the cornea, which very rare that cancer can occur there, and also from the testes of boys. We use them. There's a tissue in the testes that never get some cancer, and then we got them and we digest them by enzymes to release the cells, to open the intercellular space, and then to get the exosomes. And we have found these, like in the cornea of the chicken, we have found that they have tumor suppressor genes, that is P53. It's very well known protein that it's called the guardian of the genome, which prevents cancer from occurring in cells. So this work has been done with Dr. Alex Tendler, and we have isolated them electron microscope. We determined their size, and we found that they are indeed really anti-cancer. Before this, I just want to mention that in a healthy cell, we have two ways or two mechanisms. One is tumor suppressor genes, and the other oncogenes. We targeted two targets, P53 as a tumor suppressor genes, and KRAS, which is a gene that causing proliferation for the cells. However, if in these two targets mutation occurs, the cell just doing an arrest and either go to repair, DNA repair, and then start again to live, or they can induce the mechanism of apoptosis and kill the cell, because they don't want the mutation to go through the cells into the descendants. However, if this mutation cannot be repaired and apoptosis cannot be done or cannot be induced, so the cells turn to be cancers. Oncogenes is going up, tumor suppressor genes going down, and there is uncontrolled proliferation. and cancer is developed, and there's lots of cancer cells, and the balance is tilted because the activity of oncoproteins is much more than activity of tumor suppressor genes. So how do we do? We target the p53 by exosomes. Exosome from the retina, as I said, they have lots of p53, and they are indeed anti-cancer. And we can target the KRAS by DNA. That is what has been done with Professor Ryan, that he has developed these peptides that they can target specifically and selectively the KRAS. And we have recently published this work, and we can see that exosomes, they do some apoptosis to some cancer cell line, and eventually, actually right now, we are doing the combination of DNA and protein together, and also there's lots of constituents there. And recently I have been granted from the Ministry of Innovation and Science Technology a grant to pursue this study in pancreatic cancer, and we are doing this just for to help patients. Very important to help patients with cancer. If you can see this small girl, she wants to have hair. She just draw the hair in the mirror, and that's it. Thank you very much.
Thank you, Dr. Falax. I think that we really need more and more hope with treating those terrible disease conditions, especially when it affects children, and it is more painful than anything else. Any questions?
Thank you for your presentation. Do these exosomes exist on the plant kingdom? Or
yes, they do exist in the plant kingdom. And now we are just also isolating them from some fruits. And from fruits. Fruits. And we use them for as an antibacterial. I have another project that can be that could be an antibacterial. It's very important. I forgot to. You talked about multi-targeting. So this exosome can be you can fill it with so many molecules that you can target because all the targets, they are intercellular inside the cell. So this can go adhere to the cell. Yeah, exactly.
So what makes them different from liposomes, for example?
Liposomes, they are also organelles, but they are filled with enzymes. They are important for the degradation of cells and proteins. Like also there is a peroxisome, which has also some proteins which are also enzymes.
For plants, we are interested in plants,
Yeah.
not in human
Meat.
tissues. Okay, how easy that you can isolate them from plants?
It's very, very easy to do this. We have just some machine that can chop the fruits or the leaves or any other thing, and we can just start doing centrifugation, and the final these exosomes stay in the supernatant.
Wow.
And we can take the supernatant and do ultracentrifuge, which is about more than 100,000 Gs. So we can isolate them, and they are very stable. Like in this speed of centrifugation, even bacteria they cannot survive, but they do survive in this speed.
For a follow-up short question,
My interest is in virus, plant virus, also described, and I wonder if we can do the diagnosis on those exosomes, especially when we're looking for plant viruses. Sometimes we don't know where to enrich the tissue for diagnosis of viruses, and maybe this is one thing that's trafficked them, and might be a target that we should look into.
Yeah, very nice. We can. Let's do it after Ramadan.
Okay.
Are you actually using unaltered exosomes as your therapy, or in what way do you change?
Right now I'm using natural.
Microphone.
We can't hear you.
This field is very important. Actually, I forgot to tell you that next week there is a very important conference in Weizmann Institute for 5 days, and it's called Extracellular Vesicles. If they are friends or foes.
Thank you.